Hello there. Once again, it's Ma'am Florence and we are now at the third video lecture for cellular respiration. Earlier in the previous video, we had glycolysis and fermentation. So now we will continue. So if you notice here in the slide, we are done with glycolysis and fermentation, which is an anaerobic process. So now, let's continue to the other steps, which are aerobic. Okay, let's see. So, after glycolysis, if oxygen is present, pyruvate oxidation takes place. So, it is aerobic. So, here what happens is the pyruvate, the two molecules of pyruvate, molecules that have three carbons, are transported through the mitochondrial membrane to the matrix, and it is converted into two acetyl-CoA molecules that have two carbons. Okay, so it's like moving the pyruvate to the matrix, okay, inside our mitochondria. So if it is done, then pyruvate will be converted into acetyl-CoA. Okay, it looks like this. So look at our pyruvate here. It's in the cytosol, so it's outside, and it has 1, 2, 3 carbons. Okay, so this pyruvate will be transported. So we have a transport protein to help us do the job. And then carbon dioxide will be released. So 1 carbon will be taken. Then after, we have our NADH. So it gain an electron, NAD. Gain an electron. We have our NADH over here. So reduction happened, and then a coenzyme is added as well. So that leaves us with this product, acetyl-CoA, and it has how many carbons? 1, 2. Okay, so again, from pyruvate, a 3-carbon molecule, we are now left with acetyl-CoA, a 2-carbon molecule. So it's now inside the mitochondrion. Alright. Okay, so these are the end products of pyruvate oxidation, or it can also be called the grooming phase. So we have 2 molecules of NADH, 2 molecules of carbon dioxide, and 2 molecules of acetyl-CoA that has 2 carbons each. Okay, so after glycolysis from glucose to pyruvate, we have the pyruvate oxidation where we converted pyruvate into acetyl-CoA, right? So what's next? We have the Krebs cycle. Okay, let's see. So the Krebs cycle is also called the citric acid cycle, and the location for this is now in the mitochondrial matrix. So if we look at our mitochondria over here, the matrix is the innermost part, inside the folds, the cristae. Okay, so acetyl-CoA with 2 carbons bonds to oxaloacetic acid, or OAA. Here, OAA, which has 4 carbons to make the product citrate or citric acid. So that has 6 carbons. And since it's a cycle, it takes 2 turns of that to oxidize 1 glucose molecule. Remember, on glycolysis, the very first process, we started with 1 glucose molecule, right? So to fully oxidize that, we need to take 2 turns. Okay, so this is what it looks like, Krebs cycle. But let us simplify it. So let's look at this illustration over here. So this is 1 turn of the Krebs cycle. Let's zoom in a little bit. So you see we have our acetyl-CoA here, and only 1 molecule enters the cycle. Okay, so remember, acetyl-CoA has 2 carbons, and it will bond with oxaloacetic acid that has 4 carbons. So if they bond together, just have to add that. 4 carbons plus 2 carbons, you now have this product, citrate, which has 6 carbons. And then after that, 2 molecules of carbon dioxide will be given off. Okay, So we are left with how many? We're left with 4 carbons, right? And then afterwards, we have some reduction going on. We have our NADH over here. We also have phosphorylation because there is ATP. So there's a creation of ATP. Also, we still have reduction over here. We have FADH, so it gained an electron. And lastly, we are back with OAA. Okay, so from citrate with 6 carbons, so minus 2 since 2 carbon molecules will be released. Okay, after all of these processes, we are back with our OAA. Okay? So, I also said earlier that it takes 2 turns, right, to oxidize a glucose molecule, one glucose molecule. So, let us see how 2 turns look like. Okay, look. So, over here, we have 2-acetyl-CoA entering our cycle. So, it will bond with OAA again. So, we now have 2 molecules of citrate. So, we're just doubling the first cycle that we looked at. So, there is now 4 carbon dioxide released, 6 NADH, 2 ATP created, and 2 FADH. Okay? Okay, so in 2 turns of Krebs cycle, this is our total net yield. So, we get 2 molecules of ATP through substrate level phosphorylation, 6 molecules of NADH, 2 molecules of FADH, and 4 molecules of carbon dioxide. Okay, so in Krebs cycle, we have a lot of products. So kindly take note of these products. Okay, let's check our progress so far. Done with glycolysis, done with pyruvate oxidation, and done with Krebs cycle. Woo, we're almost done. So, I think that will be the end of this video lecture. So, please click on the last one, finally, where we talk about electron transport chain and oxidative phosphorylation. Thank you.